Hello, General Biology, and welcome to the fourth in the screencast series of Chapter 7, Cell Structure and Function. And we left off, we were talking about the chromatin, and it's the genetic material that's found inside of the nucleus, and chromatin is descriptive of how the genetic material appears at the certain stage of the cell's life, which we call a life cycle, or the cell cycle. So chromatin is actually a Greek term which means thread-like, and what happens is this chemical molecule of DNA, which we learned in Chapter 2 was in the shape of a double helix. It actually winds and coils around special proteins that are called histones. And this winding and coiling is just kind of like a slinky where you can put that 20-foot long slinky into a really tiny box when it slinks all up. Well, basically this is the same idea, but our tiny little box is going to be the nucleus of the cell. What will happen is during the replication part of the cell cycle, this chromatin will actually condense like that slinky does and form this X-shaped structure which we call the chromosome. And the chromosome is broken down into 4 parts, which we refer to as a tetrad. That's T-E-T-R-A-D, a tetrad. And in this configuration, it can very easily be split right down the middle so that when the cell divides, one half of the cell will get one half of the chromosome, the other half of the cell will get the other half of the chromosome. Half of a chromosome is referred to as a chromatid. That's C-H-R-O-M-A-T-I-D. So when we look at this structure of a chromosome, it's actually made up of sister chromatids, or 2 chromatids, and they're sister chromatids because they're actually matching, because they've been replicated. So if this is genetic information for eye color, on this chromatid, it's genetic information for eye color. If this batch of DNA is for your foot size, then this batch is for your foot size. Now the 2 chromatids, the 2 sister chromatids, are joined together in the center by a special clumping of histones and nucleosomes that we call the centromere. And the centromere is going to be used, one, to hold the chromatids together, but to attach to special structures within the cell that are called spindle fibers that are going to enable it to move from one side of the cell. So all very, very complex. But we see this chromosome when the cell is getting ready to replicate. So our genetic information can take on 3 different forms in the life of the cell. It can take on the form of chromatin early on in the cell life. It can take on the form of the chromosomes, this distinct X-shaped structure that's made up of sister chromatids, matching chromatids. And when the cell divides, then each cell is going to have an individual chromatid, half of that chromosome pair. And we'll talk about that in a later chapter, but what I need you to know is whether it's chromatin, chromosomes, or chromatids, it's all made up of this same chemical molecule, DNA. And the sequencing of that DNA determines the types of proteins that are going to be made by the cell. Okay? I hope you understand that. If not, please ask questions in class. All right, we mentioned the nucleolus, and I'll have to tell you that when I was in high school and in 10th grade, my teacher didn't know too much about the nucleolus, and she said it disappears when the cell gets ready to divide, and she told us it was responsible for forming a structure called a centriole. Well, now, not too long later, within the past 30 some odd years, we found out that she was wrong. And was she teaching me bad science? No, she was teaching me the science she knew at the time to be true. But now, with the advancements in technology and our ability to better study the nucleolus, we know a few things. One, it is not a membrane-bound organelle, but it is still considered an organelle because it serves a purpose. It has a function. We know that it's made up of parts of ribosomes, and these parts of ribosomes are called ribosomal subunits. If you've ever seen a yin and a yang, how they're shaped, that's kind of like roughly the shape of a ribosome, and they're made up of these subunits, and subunits are just parts. So there's ribosomal parts inside that are gathered together in this dense region of the nucleolus. We also know that it contains RNA nucleotides, and RNA nucleotides are just pieces of RNA. They're the phosphate group, they're the ribosose sugar, and they are a nitrogen base. And they're individual nucleotides that are floating around. We still don't know where these RNA nucleotides come from. We still don't know where the ribosomal units come from, but we know that early on in the cell's life they condense together to form the nucleolus. And we do know that when the cell is getting ready to divide and the nucleus is getting ready to divide its genetic information, that it disassembles. It can easily pull apart. And that's one of the advantages of not being a membrane-bound organelle, where it can disassemble very easily for cell division. That's going to be important because all of this chromatin is going to have to be able to move freely around, and with the nucleolus in place that won't be able to happen. So the nucleolus just breaks apart and disappears, and after the nuclear division is complete, it will reform inside of the nucleus. Okay, any questions so far about those parts of the cell? Now make sure all of that information that I told you is important, so if there's anything you don't understand, please go back and replay this, write down your questions, and ask me when we get to class. Okay. The next structures that we're going to talk about are going to be organelles that are going to be used in storage. They don't have stomachs, so they need to be able to store nutrients and waste products someplace. Also things that help clean up because there's going to be cellular activity that's happening that produces waste, so they have to be able to get rid of that waste product. And then support. Something has to hold all of this together inside of the cell. Okay, so one of those organelles is referred to as a vacuole. And a vacuole is for storage. It's going to store things like water and salts and proteins and carbohydrates. Sometimes it's even going to store digested materials depending on the type of vacuole. In this particular picture, this is a very large vacuole, and this is one that you would find in plant cells, because one of the things that you're going to have to be able to do is determine the difference between plant and animal cells. And one of the differences between plant and animal cells is that plant cells have larger vacuoles. The reason why they have larger vacuoles is to store water, but also to create something called turgor pressure, and turgor pressure helps to give that cell structure. So they will have these large vacuoles. In animal cells, the vacuoles are much smaller because usually animal cells have much more complex systems than plant cells would have, and they don't need to have that pressure within the cell with that. We also have in the protists that you saw in the communities lab, the ciliates and the flagellates, there's special types of vacuoles called contractile vacuoles that are located within them. Since protists live within water, one of the things that they have to do is be able to regulate the amount of water that enters and leaves their cell. So they have these special vacuoles that can literally contract and squeeze out excess water so that the cell won't burst and take in too much water. But that's the vacuole. Okay, there's other smaller storage vesicles or vacuoles or organelles that we refer to as vesicles. And these are tinier. And the vesicles are going to be used in transport. There's going to be things that the cell is producing, like proteins or waste products that need to be transported. So this special vesicle is a way that we can carry those materials. A lot of the vesicles carry packaged proteins. Remember I said one of the biggest things that a cell is going to do is make proteins. That's what the DNA is for. And you're going to have to be able to transport those proteins, and the vesicles do just that. Because their cell membrane structure is made up of the same cell membrane structure as all the other organelles and the cell membrane, the membranes can fuse together and kind of open up and release whatever is inside of them. In section 7.4, we'll talk about that form of transport. But here we have a vesicle that's carrying substances. This happens to be a food vesicle that has nutrients in it. And we're going to talk about a special type of vesicle called a lysosome in a few minutes that will put digestive enzymes in and help digest those food particles. Whatever does not get digested, the vesicle can attach to the cell membrane, fuse with it, and release it out. So that's one of the advantages of the vesicles. When we talk about the endoplasmic reticulum, we're going to talk about vesicles breaking off, carrying proteins, and those vesicles are going to be able to fuse with another organelle known as the Golgi apparatus. And the Golgi apparatus is going to be involved in storing and transporting proteins. Again, all linked to this proteins. So having the same membrane structure, they're able to fuse together. The storage is just temporary, and they can break off of the endoplasmic reticulum, as I mentioned, and off of another structure known as the Golgi apparatus. And when we look at the video, it's going to look kind of like a lava lamp with these vesicles breaking off. Now, you heard me mention lysosomes. Lysosomes are specialized vesicles that in particular carry digestive enzymes. So they're able to fuse with a food vesicle or a food vacuole and put their digestive enzymes in and break down the materials so that they can be used by the cell. They also have a nickname called the suicide sacs of the cell because if the cell becomes damaged in any way, they will actually rupture and destroy the cell that they're in. So that's one of the reasons why, like when you get sunburn, you've got that flaky skin because those lysosomes have ruptured and they've destroyed that cell. We used to say that lysosomes were only found in animal cells, but now notice the wording, they are primarily found in animal cells. So that means that we are finding some lysosomes in plant cells, which is new, something that I didn't teach about a few years ago. Okay. Let's stop here and we'll come back and we'll talk about the cytoskeleton in the next screencast.